Okay, so this video is for biology, chapter 13.3, about mutations. So when DNA replication doesn't go correct, or a base is exchanged or taken out or added or anything like that, or just the process of taking DNA and transcribing it into RNA, there could also be problems that happen then, or from RNA to a protein translation, it can be translated incorrectly. Any of those things are essentially mutations. A point mutation, you can add this on your notes, occurs at a single point on the DNA sequence. And that DNA sequence is part of a gene, right, like we've talked about. And most often, point mutations, where just one little point on that sequence, that mutation often occurs during DNA replication. So inside the nucleus, when the DNA is splitting, the double helix is splitting and new nucleotides are being added to each strand to make a complementary strand. That's when point mutations, usually happen. Some examples. So here is our DNA sequence, ATTAGCGA. Okay. Substitution mutation is when one base is replaced with a different base. So add in the word replace there. So instead of being ATTAG, notice in this example, ATG. So the G replaced the T there. Insertion mutation. So add in the word insertion, that's another type of point mutation, is adding a base to the sequence of DNA. So another base is just put into that same sequence. So you can see after the G here, a T has been inserted into that DNA sequence. The third type of point mutation is called a deletion mutation, and it's when a base is removed in that DNA sequence. So an example would be in our original sequence, if a T was just taken out. And if you squish it together, it would just be ATAG. Okay? Now, all three of these type of mutations, substitution, insertion, deletion, are considered, well, actually not all three. I should rephrase myself here. All three of them, or re-say that, all three of them are point mutations. But two of them, when a base is inserted or a base is deleted, it totally shifts the reading frame. It's called a frame shift mutation. We know that, like for example, look here. In insertion, we read it ATT, 3 at a time, AGT, okay? When they're transcribed to RNA, we totally shift the reading frame down to the right if you add a base. Okay, totally change the rest of essentially the reading of that instructions after you insert a base. Same with if you delete a base. It's going to totally change how that DNA sequence is read or transcribed to RNA after that point. So add in, it changes eventually then, DNA goes to RNA, right? And then is made into amino acids. So frame shift mutations will shift the reading frame, changing all the amino acids after that point. And for that reason, the insertion deletion are considered frame shift mutations. The next page just talks about there can also be chromosome mutations. This page is about chromosome or mutations on a single gene, but there can also be mutations on an entire chromosome. We're not going to get too into detail here. For biology, here's an example of a chromosome. Deletion, just an entire section is taken out. And duplication, entire section is duplicated. Inversion, essentially the chromosomes swap sides with each other. And translocation is essentially one set of a chromosome jumps to a different set of chromosomes. All right, let's talk about mutations a little bit. So mutations happen in your DNA regularly, okay? You don't even know they're happening, okay? Usually these mutations just do nothing. They don't hurt you. They don't make you better, okay? So most of the time they do nothing. Sometimes mutations can be harmful, which you're probably the most familiar with. An example, cancers can be caused by mutations, okay? UV radiation tanning beds can increase the amount of mutations that happen in your DNA, thus increasing your risk for skin cancer. Sickle cell anemia, which is a type of red blood cell disease in which the red blood cells are malformed, it's caused by a single point mutation. One letter is wrong, thus causing a lifelong disease. That's harmful. Now, you might not know that some mutations actually can be helpful. All right? So, an example in animals, mosquitoes, actually, many insects like mosquitoes eventually become resistant, or they mutate to become resistant to pesticides or chemicals that are used to kill them. Now, you might think that's not helpful, but if you think of it in terms of the mosquito or the insect, it's helpful to them because they can keep on living. Okay, so insect resistance to pesticides is an example of how a mutation can be helpful. Polyploidy is something. Oh, I'm gonna sneeze. Excuse me. Essentially, there's more. A larger amount of chromosomes in that organism, more chromosomes in it. Now, in a human, that's not going to be a good thing in our cells for the most part. Right? Well, actually always. It's usually, not a good thing. A lot of times, humans, if they have too many chromosomes in their nuclei, they don't even make it. They don't live past a certain age. And, well, they usually die preterm. But in plants that make fruit, polyploidy can be a good thing. More chromosomes, geneticists actually cause fruit to be polyploids to make the fruit larger. Why would larger fruit be a good thing? More food. So it's GM crops, genetically modified. One of the things they do is make the fruit larger, or the vegetables, so that it can feed more people. So that's mutations. Some examples, point mutations. Some of them can be frame shift. There's also chromosome mutations. Mutations can do nothing at all. They can be harmful, or they can be helpful.